Dear students, welcome to the session of biology, that is photosynthesis. So let me ask you a simple question. Have you ever wondered how the plants will take their own food materials or they can prepare their own materials? Because they don't have any kitchen, they cannot prepare, even they cannot eat the food material like animals. But the question is how the plants are able to get their own food materials. That is the very interesting one. Nothing but the plants will prepare their own food material by a unique biological process called photosynthesis. So, not only for plants, this biological phenomenon process is a basis for all other basis for the metabolic activities. That means all other organisms are depending on this process for their food material either directly or indirectly, that is. And also very interesting thing is, the another advantage for all the living organisms is the recharging of oxygen in the atmosphere. Because you know, every organism will consume the oxygen for respiration purposes, irrespective of all the things, that is either plants or animals or anything. So how this oxygen will get into the again atmosphere, that is nothing but recharging is. So another advantage for this process is, this process is also responsible for the recharging of oxygen to the environment. That is, even in ecosystems also, the photosynthesis plays a major role. That is actually we consider them as the producers. So the entire ecosystem also depends on this entire process, that is a photosynthesis. So today I am going to explain some of the key points regarding this chapter photosynthesis. That is, see, as far as the NEET is concerned, because our ultimate aim is the NEET examination, especially you are now going to write the examination in the month of May 3rd, that is. So just look once what is the frequency of questions which are asking from this photosynthesis, that is. So I am giving the data for the last 6 years here. 2020 2 questions, 2021 2 questions, 22 3 questions, 23 3 questions, 24 2 and 25 2 questions. That means on an average 2 to 3 questions are being asked continuously from the photosynthesis. So that's why also it is also important for us in the physiology chapter, that is. Now before going to that photosynthesis key points, I would explain the summary of the photosynthesis in the form of a flowchart, that is, so that at a glance you can have an idea about the entire process, that is. So basically, first concept regarding this topic is the demonstrations. That means, how are you going to demonstrate that this process takes place in the plants? Different experiments are there, variegated leaf experiment, light screen experiment, moles half leaf experiment, like this. These are the demonstrations by which we can demonstrate the occurrence of the photosynthesis in the plants. So the next important aspect is, what are the experiments which led to the discovery of this process, photosynthesis. So they are nothing but early experiment. The heading given in the NCERT textbook is early experiments. So they are starting from Priestley, Ingenhouse, Sachs, Engelmann, Van Niel, Emerson enhancement effect, Hill's reaction, even Rubin and Kamen. So these are the different early experiments through which finally we unraveled the entire secret of photosynthesis. That is, so now today we know each and every part of the photosynthesis, how the plant is going to convert the light energy into chemical energy, and that chemical energy is converted to potential energy in the form of carbohydrate, that is nothing but glucose. So this is the information regarding the early experiments. So next one is the very important aspect is, which cell organelle is responsible for the photosynthesis in higher plants I am talking. So naturally chloroplasts. So within the chloroplasts also, so which molecules are responsible for harvesting the light energy means pigments. So very, very important. So without pigments, the plants cannot harvest the light energy. So what are the photosynthetic pigments that are involved in the photosynthesis? So basically 3 categories: chlorophylls, carotenoids and phycobilins. So chlorophylls are again chlorophyll A, B, C, D, E like this. Next, carotenoids are again divided into carotenes and xanthophylls. And the exclusively algal pigments like phycobilins, two types are there, phycoerythrin and phycocyanin. So next, organization of these photosystems. How these pigment molecules are associated with the proteins in the thylakoid membranes. So finally, they are going to organize into photosystems. There are two types of photosystems. One is the PS1, the other one is PS2. It is given actually based on the nature of discovery, that is. So anyway, the details later we will see the components of the photosystems also. Next one is the mechanism of the photosynthesis. So mechanism means broadly you can divide the process into two types. One is the light reaction, the other one is dark reaction. Light reaction and a dark reaction. So actually light reaction means directly depending on the light. Here the light energy is converted to chemical energy, that is the light reaction, which occurs usually in the grana thylakoids, the light reactions. Of course, a series of photochemical activities this is. Second one is the dark reaction. So here actually the carbon dioxide is converted to or fixed into carbohydrates. That is called dark reaction, mostly enzymatic. So this occurs in the stroma of the chloroplast. So this is the two major parts of the mechanism of photosynthesis, light reaction and dark reaction. Even in the light reaction also, again, two important aspects are, one is the electron transport system, second one is the photophosphorylation. So electron transport is again divided into two types, non-cyclic electron transport and cyclic electron transport, that is. So anyway, later I will discuss the more details. So these are some of the differences between non-cyclic and cyclic electron transport. And last aspect of this light reaction is the photophosphorylation. So ultimately how the ATPs are being synthesized in the chloroplast, how the light energy is converted to chemical energy. So that is explained by Peter Michel by Chemiosmotic Hypothesis. This is the end product here. So basically the end products of the light reaction are ATP and NADPH2. These are the two types of molecules that are being formed during the light reaction. Next one is the dark reaction. So dark reaction means it is not directly dependent on the light, it is independent of light, but it depends on the products of the light reaction. So where actually the carbon dioxide is reduced to carbohydrates, that is. But again there are two different types of carbon fixation pathways are there. One is the C3 cycle, second one is the C4 cycle. Ultimately sugars are being produced. And the next aspect is what are the questions being asked in the last 5 years, that is in this chapter photosynthesis. So I am displaying some of the questions here, that is this is 2025 question. Of course I am not going to study, even answer is also written here. These are nothing but the PYQs, the previously asked questions in the NEET examination, that is. So this is another question of the 2025. This is matching. So here the question is a matching type. So next is the 24. So here another again MCQ is there, that is the question. The answer is also given here. Next one is another question in the 2024. There is also multiple options. So that means in this chapter they are going to frame the questions in different manners, either simple MCQs or multiple options or matchings or even assertion and reasons also. That's why to give awareness I am going to present these questions, that is. So this is a simple MCQ in the 2023. So next one is the PYQ of 23, another question was a simple MCQ. Next one is 23, also again simple MCQ. So these are some of the questions, 3-year questions I quoted just for your awareness. So the essence is all types of questions may be framed in this chapter photosynthesis. MCQs, multiple statements, that is matching, even assertion reason type of questions are also. So that based on that you can prepare the aspects with regard to that concept. This is the NCERT 11 latest textbook, that is. So here I underlined some of the key points where you can also do the same, because quickly you can revise the important statements in this chapter. So this is the introductory part of the plant physiology, where the two aspects is the origin of the different branches like ecology and physiology plus biochemistry. That is depending upon the level of organization. Organism or above level of organism led to evolution of a branch called ecology. That is. So next one is the cellular and molecular level of organization. That is, it leads to the formation of physiology and biochemistry. So this is the key point here. So next page. So this is regarding the scientist given, Melvin Calvin. Anyway, so I made some two key points here. One is the Calvin proposal of the change light energy to chemical energy by transferring an electron in an organized array of pigment molecules. This is one. Second one is the principles of photosynthesis as established by Calvin are at present being used in studies on renewable resource for energy and materials and basic studies in solar energy research. So these are the two important aspects, that is in the scientist. Now coming to the chapter, actual chapter, the photosynthesis in higher plants. That is, so here the nutrition concept, autotrophic nutrition and heterotrophic consist. That is, anyway, autotrophic means they can prepare their own food materials. That is, all green plants you can say definitely autotrophs. That is, and it is being carried out by a physico-chemical process, that is photosynthesis. So next one is it is the basis of life on the earth. Already in the introduction I gave you. So what are the two reasons? Means it provides the oxygen to the entire atmosphere, recharging of oxygen. That is, second primary source of all food on the earth, even including the ecosystems also. So this is the next demonstrations, variegated leaf experiment, where you can demonstrate the chlorophyll is essential for photosynthesis, light screen experiment, where light is essential for photosynthesis, and moles half-leaf experiment, that is CO2 is essential for photosynthesis. So these are the three demonstrations what I made actually, that is. Next, coming to the early experiments which led to the discovery of the process photosynthesis. This first one is Joseph Priestley concept. That is, he used the mint plant. So bell jar he used. So his statement is plants restore to the air whatever the breathing animals and burning candles remove. So by which we came to know nothing but the purification of the air by the very first experiment by Joseph Priestley. So next one goes to, that is another scientist, John Ingenhouse. So Ingenhouse, two important aspects are there. That is, so one is the only green plants can purify the air is the first one. Second one is, and the light is essential for photosynthesis. In presence of light only it can purify the air. The two aspects which were given by John Ingenhouse. So next scientist is Julius von Sachs. So that is mainly two aspects. Glucose is usually stored as a starch, that is, and chlorophyll, the green pigment located in the special body is called chloroplasts. The two aspects regarding the von Sachs. Next one is the first action spectrum provided by Engelmann. So Engelmann used the plant material Cladophora, multicellular alga, Cladophora, and aerobic bacteria also he employed. So finally what the outcome is, that is first action spectrum of the photosynthesis described, and here the photosynthesis mainly occurs at blue light and red light. This is the experiment of Engelmann. Next one is the microbiologist Cornelius van Niel. That is, so his contribution is definitely, his identification is there is a molecule which is a hydrogen donor for this process. The hydrogen donor is there by which carbon dioxide is reduced to carbohydrate. So he proposed that in green plants water is the hydrogen donor, but in case of the bacteria, that is, the hydrogen donor is hydrogen sulfide instead of the water. That's why In higher plants, the byproduct is oxygen, but whereas in bacteria, the byproduct is sulfur or sulfates. This is the contribution by Van Niel. That is, based on this, you can identify oxygenic photosynthesis and anoxygenic photosynthesis. Generally, we can. Here, the blue-green algae also show oxygenic photosynthesis. That is important aspect here. So, next point is, where does photosynthesis takes place? So, that is, can you name some other parts where you means chief organs are leaves, young stems, that is, so sepals, like wherever green parts are there, there you can see, that is. And this is the regarding alignment of chloroplast based upon the light intensity. So, in high light intensity, what happens is the chloroplasts are aligned parallel to the radial walls, and in low light intensities, they are perpendicular to the radial walls. That is the concept. So, next, anyway, this is the light reaction, dark reaction already given. This is the diagram, that is. So, next important aspect is the first one is this is the absorption spectrum of the various pigments, that is. So, before that also a small experiment is there. The leaf pigments of green plant can be separated through paper chromatography technique. In higher plants, 4 types: chlorophyll B, chlorophyll A, keratins and xanthophylls. This is the technique, just you remember, paper chromatography technique. And colors also important. Chlorophyll A is bright or blue-green, chlorophyll B is yellow-green, xanthophylls yellow, and carotenoids yellow to yellow-orange. These are the colors actually. And this is the figure where absorption spectrum of different pigments. So, this is the blue one is the chlorophyll A, that is chlorophyll A. So, chlorophyll A has absorption maximum at blue and red, 2 peaks are there. Same in case of the chlorophyll B also the same case. High absorption at blue light and red light, and whereas this is the carotenoids. So, next one is the action spectrum, that is. So, action spectrum means the graph indicates the rate of photosynthesis at a different wavelengths, that is. So, different wavelengths, again the peaks are blue and red only. So, highest rate of photosynthesis can be shown in blue light and red light. So, this is the next one. Chlorophyll is the major pigment responsible for trapping the light. Of course, the others are, that is carotenoids also. They are called accessory pigments. The carotenoids can be considered as, because they not only enable a wider range of wavelength incoming light to be utilized for photosynthesis, but also protect the chlorophyll A from photooxidation. That's why you can also consider them as accessory pigments or shield pigments also, that is. So, next one is the photosystem, that is. So, this is the structure of a photosystem where you can divide it into 2 types, PS1 and PS2. In both cases, the core is called reaction center. It consists of a special chlorophyll A molecule bound to the proteins and surround the different types of pigment molecules which will form light harvesting complex or antenna. So, from the reaction center, directly it is electron is being accepted by primary electron acceptor. Anyway, for PS1, the reaction center is called P700, but for PS2, it is P680. That is based upon the amount of the light absorbed at different wavelengths. That is the, so here the given PS1, P700, PS2, P680. That is the structure of photosystems. Next, this is the non-cyclic electron transport or Z scheme. So, involvement of 2 photosystems here, that is. So, electrons then are moved downhill again, this time to a molecule of energy rich NADP. Ultimately, the electron will go to NADP plus. So, ultimate donor is water, final electron acceptor is NADP plus, that is. So, why is it called Z scheme? Due to its characteristic shape. The shape is formed when all the carriers are placed in a sequence on a redox potential scale. That's why it is also known as Z scheme. Next, photolysis of water, splitting of water, that is. So, the splitting occurs on the inner side of the membrane towards lumen, thylakoid membranes. So, here this is the concept actually. and the water molecule splits into 2 protons, 2 electrons and half O2. This is photolysis of water. Of course, 2 water molecules he mentioned. That is. So, it is assisted by OEC, oxygen evolving complex, that is located on the inner side of the membrane of the thylakoid, that is. Next one is the photophosphorylation. This is cyclic electron transport. Cyclic, that is. So anyway, phosphorylation concept is either in mitochondria or chloroplasts. In both cases, ATP synthesis occurs called phosphorylation. But in case of the chloroplast, the source of energy for the ATP synthesis is light. That's why it is called photophosphorylation. But whereas in case of the mitochondria, the source is oxidation of the high energy molecules. Hence, it is called oxidative phosphorylation. But anyway, in both cases, ATP synthesis takes place. That is. So in non-cyclic, you can see both ATP and NADPH2 are synthesized. But whereas in cyclic electron transport, only ATP synthesis takes place. That is. So generally, grana thylakoid contain both PS1 and PS2. Hence, the non-cyclic occurs. But whereas in stroma lamellae, usually it lacks PS2. No OEC. That's why cyclic electron transport occurs in the and cyclic photophosphorylation also occurs when only light of wavelengths beyond 680 are available for excitation. And the most important concept is the chemiosmotic hypothesis. Peter Michel is the scientist who provided this chemiosmotic hypothesis to explain the ATP molecules, and he got Nobel Prize also for this one. And basically 4 aspects are there. One is the membrane, second one is proton pump, third one is proton gradient, and last one is the ATP synthase CF0CF1. That is, this is the chemiosmotic theory. Anyway, this is also the diagram explaining the chemiosmotic theory. That is, the concentration of the proton is always very high in the lumen, that is, and very low in the stroma. So when protons are moving from higher concentration to lower concentration, this is facilitated diffusion through CF0. So they release some amount of energy in the head by which ADP is converted to ATP. This is the photophosphorylation, that is chemiosmotic hypothesis. Primary electron acceptor of the PS1 is the outer side of the membrane, transfers electron to electron carrier but proton carrier, the hydrogen carrier, and both proton plus electron here. Next, NADP reductase enzyme is located on the stroma side of the membrane, important. That's why the NADPH2 synthesis takes place on the membrane of the thylakoid towards the stroma side, that is. And anyway, for this gradient, the protons are removed from the stroma only. And next, lumen contain more protons than stroma, that is in terms of pH also is explaining. So lumen generally contain very low pH. You know, hydrogen concentration, proton concentration is very high. Low stroma contain high pH, that is. Next, the gradient is essential for the breakdown of, gradient is responsible for synthesis of ATP molecules. Next, ATP synthase enzyme structure is explaining. CF0, CF1. CF means coupling factor. CF0 is also called stalk. CF1 is also known as head. Next, these are the 4 parts already explained you. Chemiosmosis requires a membrane, proton pump, proton gradient, and ATP synthase. That is. So this is regarding the light reaction. Next comes to dark reaction. The question is where these ATPs and NADPH2 are produced in the light reaction are used. Actually, the ATP and NADPH2 considered as assimilatory power. So these high energy products are used in the dark reaction actually, that is. So here the dark reaction, already told you 2 types of cycles, C3 cycle and C4 cycle, that is. So in Calvin cycle, the first stable product that is formed 3-phosphoglyceric acid, a 3-carbon compound, hence it is called C3 cycle, that is actually. Whereas in C4, the first stable product is OA, oxaloacetic acid. So here it contains 4 carbons, hence it is called C4 pathway, that is. Next, coming to the Calvin cycle, so primary acceptor of carbon dioxide is RUBP. RUBP, a 5-carbon keto sugar, ribulose 1,5 bisphosphate carboxyl, RUBP. ribulose 1,5-bisphosphate, that is keto sugar actually. So you can divide the Calvin cycle into 3 steps: carboxylation, reduction, regeneration. So carboxylation is a very crucial step of the Calvin cycle, because where CO2 is utilized the carboxylation of RUBP, catalyzed by rubisco, the most abundant protein in the biosphere, that is. So this is the diagram of the Calvin cycle. So this is the carboxylation part, this is the reduction part, regeneration. Finally again you are supplying the RUBP for the cycle process. Next one is the reduction process. So reduction means hydrogen source. So who is hydrogen source means NADPH2 that is formed in the light reaction. NADPH2, that is the reduction process. So for every 1 carbon dioxide reduction, what is the assimilatory power in the Calvin cycle means 3 ATP and 2 NADPH2. Or what is the assimilatory power required to produce one net glucose, that is 18 ATP and 12 NADPH2. That is called assimilatory power. So this is the Calvin cycle. So next one is the C4 pathway, that is. So C4 plants contain a special anatomy called Kranz Anatomy, that is. So here the vascular bundle is surrounded by thick-walled bundle sheath cells without intercellular spaces, and they contain agranule chloroplast. That means chloroplast dimorphism occurs in C4. The reason is the division of labor, because some dark reactions occur in the mesophyll cells, some dark reactions occur in the bundle sheath cells. That is basically Kranz anatomy significance. Of course, this another significance of the Kranz anatomy is to enrich the CO2 concentration to suppress the photorespiration in the bundle sheath cells. That is. So here the primary acceptor of CO2 is phosphoenol pyruvate, 3-carbon organic acid, and first stable product is OAA. That is. So this is the diagram. So this is the mesophyll cell, this is the bundle sheath cell. So actually C4 also contains Calvin cycle, C3 cycle. So the Calvin cycle occurs in the bundle sheath cells, not in the mesophyll cells. That is. So this is the diagram. So next one is the photorespiration. It's a biological waste process, that is, and this can be seen in the C3 plants, generally not detectable in the C4 plants. I will explain in the next video, that is C4 level, that is. Rubisco is the most abundant enzyme in the world biosphere. In C3 plants, some of the O2 does bind to rubisco and hence CO2 fixation is decreased. That is the reason for the photorespiration. That is. So here what happens is, in presence of high light intensity and high oxygen concentration when compared to CO2 concentration, the rubisco is binding to, RUBP is binding, rubisco act as oxygenase. So hence RUBP reacts with oxygen and release the carbon dioxide. So in this process, there is no synthesis of ATP, no sugar, but at the same time, the loss of fixed carbon is there. That's why it is a biological waste process, that is. Generally, these plants show some tolerance to higher temperatures. This is another key point here. So next one is the differences between C3 and C4. Already in this process, we discussed all these points, but anyway here directly given in the tabular form, that's all. And the last aspect here is the factors affecting. So factors means there are two types, external factors and internal factors, that is both. So external factors are light, temperature, carbon dioxide concentration, oxygen concentration and water. These are the important. Coming to the internal factors or plant factors are number, size, age and orientation of leaves, mesophyll cells and chloroplasts, internal CO2 concentration and the amount of chlorophyll, that is. Anyway, this is the Blackman's law of limiting factor. He proposed the law of limiting factors. And when a biological process is controlled by several factors, out of all the factors separate, the lowest factor is called as, that is, lowest factor is called as limiting factor, that is. But anyway, I will explain the next video. A short video is there I can explain. So this is the carbon dioxide concentration. This important here is saturation of the C3C4, frequently asking. So here C4 get the saturation at 360 microliter per liter, whereas C3 it occurs beyond 450 microliter because photorespiration occurs in C3, photorespiration doesn't occur in C4. That is a reason actually. So the commercially, how are you going to suppress the C3 crops means by growing them in the greenhouses where CO2 concentration is more. Example, tomatoes and bell pepper, anyway he mentioned. So next one is the temperature. Generally temperature is of course genetic, that is, and water. Here important point is water is of the reactants in the light reaction, no doubt about it, donor of hydrogen. At the same time, its effect is more through its effect on the plant rather than directly on photosynthesis. So that is the advantage, that is the point of water actually. So this is actually the textbook. So try to consolidate some of the key points here, that is. Okay, now just I will give you the brief videos explaining the concepts. The first video is, this is non-cyclic electron transport or the jet scheme. So the basic point is involvement of two types of photosystems, PS2 and PS1, both are involved here. So it requires shorter wavelength of red light and longer wavelength of red light also, that is. So this is the PS2 first initiation, this is the electron acceptor, then electron transport system goes to LHC, finally primary electron acceptor of the PS1. Ultimately the electron is, final electron acceptor is NADP+, it is reduced to NADPH2. Then who is ultimate donor of, that is, electron means water is the ultimate donor of electrons in this case, that is non-cyclic. So why is it called jet scheme means, if you plot these factors based on the redox potential value, the path is zigzag, that's why it is called jet scheme concept, that is non-cyclic electron transport. So in the non-cyclic electron transport actually both types of assimilatory power are being produced, ATP as well as NADPH2, both are produced. That is the advantage of non-cyclic electron transport or jet scheme. So in the next diagram I think is more. So this is the already textbook, in the textbook you mentioned here, this is the non-cyclic electron transport. So this is the PS2, this is the PS1 and cytochrome B6 subcomplex. So this is the photolysis of water occur in the lumen on surface of the inner membrane towards the lumen, and NADPH2 formation takes place on the outer surface of stroma, granothylakoid, that is NADPH2, that is the formation. This is another diagram. So this is another diagram of non-cyclic more clearly. Of course, these questions may not be asked just to have an idea. So basically these are how are they aligned in the membrane. That is, this is the PS2, this is photolysis of water, cytochrome B6F complex, plastocyanin, a copper containing mobile carrier. Next one is the PS1, and from PS1 it goes to NADP+, that is. As a result, what happens is more proton concentration occurs in the lumen than stroma. So ultimately this is photophosphorylation. So protons move from higher concentration to lower concentration, meanwhile ATP synthesis occurs on the head, that is. So this is the non-cyclic electron transport or jet scheme. So next one is cyclic electron transport. So why is it called cyclic means, the electron released from the P700 will travel through various carriers and finally reach the same molecule. Hence it is called cyclic. So here what happens is, no photolysis of water, no oxygen evolution, that is no NADPH2 formation, and involvement of only PS1, no PS2, that is. But here only proton gradient is established, so it is useful to produce only ATP molecules, no NADPH2 production in case of cyclic electron transport concept. So this is the cyclic electron concept. That's same cyclic in the original manner. This is the membrane, thylakoid membrane, that is. So this is the PS1, travel through cytochrome B6F complex and release and finally reach the same molecule, that is. So next one is the C3 cycle or Calvin cycle. So already I mentioned three key phases: carboxylation, reduction, regeneration. So primary CO2 acceptor here is ribulose 1,5 bisphosphate. a 5 carbon sugar actually. That is, first stable product is 3-phosphoglyceric acid and no assimilatory power is required. Important. No assimilatory power is required for the production of 3-PGA. That is, then 3-PGA undergo reduction. The donor of hydrogen is NADPH2. So, triose phosphates are produced. Sugar is being produced. This is the final product of photosynthesis actually. But at the same time, we have to provide the RUBP again to continue the cycle, for which regeneration phase is required. In the regeneration again ATP involvement, ATP requirement is there. In the regeneration, ATP molecules are required. So finally RUBP is formed. This is actually shortcut. If you want, I can give you the detailed one also, just here. So here, I consider 3 carbon dioxide molecules at a time, that is here. So first stable product is 3-phosphoglyceric acid, 6 molecules, 1,3-biphosphoglyceric acid, and reduce it to 6 trioses, out of which 1/6 will be exported to the cytoplasm for the sugar synthesis. Remaining 5/6 of triose phosphates are again converted to 3 molecules of RUBP. This is the regeneration phase. Of course, NCERT it is not there, just have an idea. So here, the unit assimilatory power for the C3 cycle is, unit assimilatory power means reduction of 1 carbon dioxide, 3 ATP molecules and 2 NADPH2 molecules, and assimilatory power for the production of 1 net glucose is, that is 18 ATP and 12 NADPH2. That is the C3 cycle or Calvin cycle. Here you won't find any division of labor because all the reactions occur in the mesophyll cells only. That is. Next one is the C4 cycle. It is also called Hatch-Slack pathway because it was discovered by 2 scientists, Hatch and Slack. That's why it is also called Hatch-Slack pathway. And first stable product formed is 4 carbon compound, that is oxaloacetic acid, OAA. That's why it is called C4 cycle. So here division of labor. Some dark reactions occur in the mesophyll cell, some dark reactions occur in the bundle sheath cell. So this is to avoid the photorespiration concept. How means? Actually the Calvin cycle takes place in the bundle sheath, not in the mesophyll cells. So what happens here is, continuously the malic acid undergo decarboxylation and release the carbon dioxide. So CO2 concentration is always very high in the bundle sheath. So under high CO2 concentration, the Rubisco act as carboxylase but not oxygenase. So that's why photorespiration is not detectable in the C4. This is the advantage. Because Rubisco enzyme is present in the bundle sheath cell, agranule chloroplast. So Calvin cycle means involvement of both C3 and C4. So ultimately sugars are being produced by C3 only. Here also, C4 cycle. But here the another important difference is unit assimilatory power is 5 ATP and 2 NADPH2. And assimilatory power to produce 1 glucose, net glucose is, that is 30 ATP and that is 30 ATP and 12 NADPH2. That is, this is the C4. That's why C4 plants are more productive than C3 plants. That is, and of course they are also evolved mainly to tolerate high temperatures when compared to this is C3, and water use efficiency is also more in C4 when compared to C3. That is. So this is another version of the C4 cycle where clearly which reaction takes place in cytosol and which reaction takes place in the chloroplast also, just diagrammatically. That is. So this is regarding the C4 cycle. That is. So next one is the photorespiration or C2 cycle. Photorespiration or C2 cycle. Why is it called C2 cycle means, here 2-phosphoglycolic acid, 2 carbon compound is a substrate, that's why it is C2 cycle anyway. So here the involvement of 3 cell organelles, chloroplast, peroxisome, mitochondria. So here that is RUBP reacts with oxygen instead of CO2. So 2 molecule, 3-PGA plus 2-phosphoglycolic acid formation, and glycolic acid again converted to 2-phosphoglycolic acid converted to glycolic acid, and glycolic acid enter the peroxisome, again oxidized glyoxylic acid is converted to glycine enter the mitochondria, two glycine molecules are converted to one serine where actually the CO2 released in the mitochondria. Serine goes to peroxisome, converted to hydroxypyruvic acid, and again it is converted to glyceric acid. Finally, glyceric acid goes to chloroplast. By taking one ATP again convert 3-phosphoglyceric acid. So here the already fixed carbon is converted to released into the atmosphere. That is the loss actually. That's why C3 plants are less productive when compared to C4. This is the photorespiration. So very brief videos these are. So this is structure of photosystem. So any photosystem contain two parts. First one is the core. Core is called reaction center, and here the special chlorophyll A molecule binds to proteins, and surrounding different types of molecules, chlorophyll A, B, keratins and xanthophylls, they act as antenna or light harvesting complex. And of course, finally when excitation takes place, electron is accepted by primary electron acceptor. So PS1 reaction center is called as P700 because this chlorophyll A molecule will absorb longer wavelength of radiate maximum. And LHC1, light harvesting complex 1, same for PS2, the reaction center is called P680 because this chlorophyll A molecule absorbs most of shorter wavelength of red light, that is, and LHC2, the antenna is called LHC2. So this is the photosystem structure, that is. This is also the same, of course, photosystem. And this is absorption spectrum, already explained you. Chlorophyll A, chlorophyll B and carotenoids. So chlorophyll A and B in both cases, the maximum absorption peaks are at blue light and red light. Absorption spectrum actually, that is. Of course, this is C3, C4 plant. Already we discussed all these points, that is. So this is the Blackman's law of limiting factors. So here if you consider the two factors comparison, that is, for example, one is the light, the other one is carbon dioxide. So initially when the light is 0, there is no photosynthesis. So if you increase the light intensity, the rate of photosynthesis also increases because at that time light is the limiting factor. This is A. Up to this point, light is the limiting factor. But beyond this B point, you see here, light is not the limiting factor because it is concentration, the intensity is very high. CO2 becomes limiting factor. So out of all the factors supplied, whichever factor is supplied low concentration, that will act as limiting factor. This is the Blackman's law of limiting factors, that is. So this is overall photosynthesis concept. But anyway, for information, after the listening this video, you can also solve some of the questions here. That is, so I gave some questions here. That is, so you try to solve them, these questions also. Okay. So thank you everybody, and hope, I think you can have the essence of this chapter photosynthesis. Thank you.